Good morning, everybody. I'm just going to wait a minute for everybody to enter the webinar and then we'll make a start. Okay. So, good morning, everybody. Thank you for joining today's webinar. I would like to introduce George Burghel, who is presenting today an overview of the recently published best practice guidelines for analysis and reporting of microsatellite instability in solid tumours. George is a consultant clinical scientist and is deputy lead for the Cancer Genomic Services in the Northwest Genomic Laboratory Hub based with us in Manchester. George also holds an honorary senior lectureship in the Division of Cancer Sciences at the University of Manchester, where he contributes to academic teaching and research in cancer genomics. George is an assessor with us for EMQN, and we are very grateful for George's time today. It's really a great opportunity to ask any questions about the new guidelines. So before we begin, I just have a few housekeeping points. This webinar is being recorded. We will share a link of the recording with all participants afterwards. If you have any questions, you can ask these throughout the webinar using the Q&A tool, which is at the bottom of the Zoom screen. We will answer all questions at the end of the presentation, or if we don't have time, we can answer by email. You can email us at office@emqn.org. This is a CPD certified webinar, and we will send certificates to all live attendees after the webinar. This may take a little time, so just bear with us. It might be a couple of weeks. Okay, with that, I will hand over to you, George. Thank you.
Thank you, Vic. So, yes, as Vic mentioned, we're covering the best practice guidelines for analysis and reporting of microsatellite instability in solid tumors. So I will start with a bit of background, why we've done this work, and then go through the recommendations. I will only be highlighting the recommendations, so I would highly recommend that you read the actual guidelines if you haven't done so already. So just for those who are new to MSI testing, MSI is basically the accumulation of insertion and deletion variants in short tandem repeat DNA sequences, or known as microsatellites. So that's the name of MSI, comes from the instability of the accumulation of these insertion deletions in the microsatellites that are short tandem repeat DNA sequences. Now, usually DNA mismatch repair system promotes high fidelity DNA replication by detecting and repairing any mismatches in the genome. MSI occurs if we lose the function of the MMR system. And that can happen for many reasons, including loss of function, pathogenic variants in MLH1, MSH2, MSH6, and PMS2. And the loss of this MMR is associated with Lynch syndrome, which I'm sure most, if not all of you, are very familiar with. And Lynch syndrome is responsible or occurs in many cancers, including colorectal and endometrial cancers. So MSI testing is increasing a lot in the clinical services. And the reason for that is that these mismatch repair deficient tumors are known to respond better to immune checkpoint inhibitor therapy. And also importantly, doing the MSI test help or inform Lynch syndrome screening. There are few guidance that exist. There is the original NCI guidance published in I think the publication was 1998 and 2004, but there were the 1996 guidance and the 2002 guidance. and they essentially provided the initial guidance. However, since then, a lot of advances in knowledge and technology happened that they are relatively out of date. Then we also have guidance from ESMO and CAP ASCO, which have been published more recently. However, they focus on recommendations with relation to immune checkpoint inhibitor therapy, and there are no specific recommendations on the MSI analysis itself. And so we felt that there was a need for a more current and comprehensive best practice guidelines. And so that was our aim when we set out to do this. We wanted to create up-to-date recommendations for the analysis of MSI that is helpful to assess solid tumor mismatch repair status. And we wanted to focus on both immune checkpoint inhibitor therapy and Lynch syndrome risk. And we also wanted to provide clarification of terminology and review some of the common methods that are used for MSI analysis. We wanted to be focused, so we limited our guidance to the MSI caused by the 4 core MMR genes, MLH1, MSH2, MSH6, and PMS2. And we did not cover the MSI analysis of, for example, circulating free DNA or used in cancer surveillance or the diagnosis of constitutional mismatch repair deficiency. So what we did to start with, we developed a working group that included 10 UK and European expert scientists, both from clinical and academic centers. We also had 2 brilliant EMQN representatives helping us throughout, and we met regularly between the end of September 2022, working through the guidance all the way to November 2024. We then submitted the guidance to European Journal of Human Genetics at the end of last year, and it was published in August 2025. So the way we divided ourselves is we had a methodology working group covering all the technical sides around MSI testing that was led by Richard and many of the other scientists. And we also had a reporting group focused on the interpretation and reporting, especially in a clinical setting. And we were supported throughout by EMQN. So big thank you to Mel and Katie for their hard work throughout. And so during our meetings, what we, and in between, we did a review of the literature and discussed important points. We discussed the definitions of MSI and all the terminology that's used around it. We also reviewed the common MSI assays. And then after we did all of that, we came up with some proposed recommendations. We divided the recommendations into MSI analysis methodology. So that's covering things like design, selection, validation, and MSI reporting, which covers both interpretation and reporting. And we structured the recommendations with 3 levels. A must, where it is essential for best practice, a should, where it's highly advised, and a may, where it's advised when or where possible and practical. And when we had our draft guidance ready, we sent that for a community consultation that happened in October 2024, which basically covered a large number of scientists participating in the EMQN EQA schemes for MSI. I'm sure many of you are here today. We also sent it to the members of the Association of Clinical Genomic Science in the UK, and we selected experts in the field from both pathology and oncology to give us feedback on the guidance. So now I will start with the guidance itself, starting with our terminology review. Now, the terms that are used around MSI analysis and outcomes vary a lot in the literature. And errors in the terminology are common. Therefore, it was important for us to start by clarifying and agreeing on the terminology. And we wanted to clarify that when we talk about MSI, we should only use that to describe the analysis of somatic insertion deletion variants in microsatellites, and not to use that term for alternative technologies to identify MMR. And when we talk about MMR, whether deficient or proficient, we are referring to the underlying pathology or biology of the tumor. So you can indeed use MMR status to describe the result of MSI analysis, but you can also use that to describe any other method that can be used to assess MMR function. So that would include immunohistochemistry of the MMR proteins or MMR gene sequencing. Then we looked at the descriptors, and we wanted to, again, limit and unify the descriptors that are used to describe the MSI testing itself. So when we say MSI high, that is reflecting using MSI test and then getting a result that's showing a significant increase in microsatellite instability. And that in itself can indicate mismatch repair deficiency. When we say microsatellite stable, that's when we refer to no evidence of instability. So there's no evidence of microsatellite insertion deletion variants. And this result indicates that the tumor is mismatch repair proficient. And there are the status of low or indeterminate, and those refer to basically uncertainty in the MSI test result, which can be because of many reasons. So moving on from determining or agreeing the terminology, we move to review the common MSI analysis methods. There's more in the guidance itself. I'm going to focus on two of them. So fragment length analysis was actually the first method to detect mismatch repair deficiency. And essentially, as many of you, again, will be very aware, you use PCR amplicons for the microsatellites. They get size separated by electrophoresis and then visually inspected, most commonly using these electropherograms, for example, where you look at them and decide whether a variant is unstable or not. In 1996, the National Cancer Institute, when they produced the Bethesda guidelines to unify Lynch syndrome screening, they recommended specific MSI markers. And these were a set of 2 mononucleotide repeats and 3 dinucleotide repeats. This was then revised in a second meeting where they produced the revised guidelines, and they moved into endorsing a panel of 5 quasi-monomorphic mononucleotide repeats, and that became the panels. There was also 2 pentamarkers that were also included. Now, as many of you will experience, fragment length analysis is very cheap and it's relatively fast. However, again, as many of the expert attendees today will know, that you do need expert interpretation of the results, and that can be open to subjective interpretation. I've been in many situations where myself and someone else will look at the results and not totally agree on whether we're describing them as microsatellite, unstable or not, and so on. There are also sensitivity issues with the technology, and sometimes you may require matched normal DNA to resolve a result. More recently, we started seeing more of the next generation sequencing assays being developed. And there's a huge variety of those, differing with library preparations, bioinformatics pipelines, and the algorithms they use to classify the MSI status. There's a huge variety in the markers that can be used. You can start from a few select ones all the way to hundreds of thousands across the genome. And essentially what it does, it compares allele distributions between the sample to a reference distribution from either a microsatellite stable control or a matched normal sample. More commonly than the fragment length analysis, you get a dichotomous outcome, whether it's MSI high or MSS. However, you still get MSI indeterminate in certain samples. The accuracy of the method depends on the sequencing method that you used and the analysis pipelines. But if you focus on a small panel, you probably have similar cost and turnaround time to fragment length analysis. You can still use very large panels, which has advantages. However, it comes with higher cost, longer turnaround time, and a higher data burden. And of course, to be able to do this for next generation sequencing, more specialist labs are needed to perform the test. The massive advantage of using an NGS-based method is that it's fully automatable, which can give you a really high throughput testing. Now, we did not want to produce the guidance without also touching upon MMR immunohistochemistry, because that's a massive alternative and complementary biomarker for mismatch repair status. However, because they are essentially measuring different things, discordance between them is expected, and it happens. We know that. We know that around 5% of mismatch repair deficient tumors may retain MMR protein expression on immunohistochemistry. I've put a couple of the references there. But we also know that if you are using an MSI test, you could well get a microsatellite stable or microsatellite instability low result when there is actually MMR deficiency. And that's been published in endometrial cancer, and also published with MMR deficient tumors that are caused by MSH6 loss. And I'll cover a bit of that in a little bit more details later on. So they are complementary. They are both different biomarkers for MMR status. However, they are measuring different things, and you can get discordance between them. So now we come to the recommendations themselves. And what we did is we divided, as you've seen when I was talking about the working group, we had two working groups between us, one focused on methodology and one focused on interpretation and reporting. So we divided our recommendations in that set. We had the methods recommendations, which refers to the entirety of the MSI assay being used. We're covering the composition of the MSI markers, the technology used to target or amplify these markers, how you visualize the microsatellite variant, and the classification thresholds and algorithms. And when it comes to reporting or interpretation and reporting of the results, the recommendations are there to aid the reporting of the MSI results with relation to the patient's risk of Lynch syndrome and also their predicted responsiveness to immune checkpoint inhibitor therapy. The interpretation or the interpretation recommendations also cover the interpretation if you have other clinical or pathological information available, such as MMR immunohistochemistry. We also have a flow chart fabulously designed by Richard, which essentially takes you on a journey that follows the lab workflow. It starts from the technical aspects, including the assay limitation itself, the sample characteristics, and then moves into results interpretation and reporting, including when MMR immunohistochemistry is available and relevant. So next, I'm going to start highlighting the recommendations. Like I said at the start, I will not repeat all the recommendations in details. I just want to highlight various parts of the recommendations, and hopefully that will give you a taster that you will go download the open access publication and adopt it or start using it in your lab. So starting from the methodology recommendations, recommendation one is to, or the first set of the recommendation is to do with methodology, and recommendation one is talking about the MSI markers. And if we are choosing MSI markers, we need to be careful to make sure to assess their constitutional variation in the genome with specific attention to ethnic background. Okay, but if you decide that for your technology of choice you want to use polymorphic markers, then you have to keep that frequency in mind, and we think that you must use paired tumor normal DNA analysis. And it's important to know that even if you're using quasi-monomorphic mononucleotide markers, which are commonly used with tumor only, you need to keep in mind that they can have high frequency in certain populations. And sometimes you may still require normal sample to exclude constitutional variant. If you are using polymorphic marker and you don't have tumor normal DNA pair, then this should be caveated as recommended in recommendation 8, which basically covers the assay limitations and the implication for interpretation. So if you don't have a matched normal tumor for polymorphic markers, this must be caveated and reported. We also recommending that assays should only use mononucleotide repeat markers. And as we mentioned earlier, sometimes you may get MSI stable or low results with MMR deficient tumors caused by MSH6 deficiency. And that's because MSH6 is mainly responsible for detecting single nucleotide mismatches. So if you have an MSH6 deficient tumor, this may only show by instability of mononucleotides. And if you are not using mononucleotides in your assay, or there are only a few of them, that could impact the sensitivity, especially for MSH6 deficient tumors. And that again should be clearly stated and caveated as per recommendation 8. Now, for the third recommendation, we recommend that you must be using a panel of multiple markers, and a minimum of 5 markers is needed to report reliably. If you are using less or reporting with less markers than that, then again, that should be clarified and caveated as per recommendation 8. Then coming to recommendation 4, assays should be validated for the tumor type being tested. Now, the original MSI development was focused on colorectal cancer. Okay, and that's the original NCI guidance and the revised version. The focus was colorectal cancer, but we now know, and the literature shows, that the frequency and the size of MSI variants can differ between different tumor types. For example, MSI encoding DNA sequences can mutate at different frequencies due to tissue-specific selection pressure. So assays should be validated separately for the tumor type that you want to report on. And if you are using the assay without being validated for that tumor type, that again should be caveated and reported as per recommendation 8. Now moving on to recommendation 5. When you're doing your validation, we do not recommend. So we say that you must not use neither tumor mutation burden or mutational signatures as a reference method when you're validating an MSI assay. High tumor mutation burden, for example, is not specific for MMR deficient tumors. It can be caused by other biological factors. And if you move to mutational signatures, they are defined by patterns of somatic variants that can again be caused by different mechanisms during tumorigenesis. And there are actually more than one mismatch repair deficient mutational signature available. They can become a really good assay in the future, but they do require large panel testing. So because of the important difference in biological causes behind them, we say they must not be used as reference method. If you are using a reference method suitable, for example, IHC, and you get a discrepant result, then you may need to think about an alternative method to use. Now, again, like we said before, discordance is acceptable. There are biological and technical differences, and there is no actual gold standard for MSI analysis. A third technology or a third alternative test may not always be available. So sometimes you may require second opinions, for example, where analysis could be subjective, or repeat testing if you still have a discrepancy. Also, when you are developing your assay, if you are choosing MSI markers from different assays, they should not be assumed to have an equivalent sensitivity and specificity. So when you select MSI markers, they should be selected for the technology that you are using them for, rather than an assumed because they work well in NGS sequencing, they will work for my technology correctly. That should not be done, and they should be selected for the technology that you are using. We now move to the reporting recommendations. So these recommendations overall aim to aid reporting of your MSI results in order to inform patients' risk of Lynch syndrome and responsiveness to immune checkpoint inhibitor therapy. And that, of course, is applicable over several tumor types. So starting from recommendation 9, reports of the MSI analysis must include a comment on the neoplastic cell content and any alternative histology, for example, dysplastic instead of neoplastic, of the sample if that's the case. All of these, neoplastic cell content and alternative histology, can have implications on the clinical interpretation. So, for example, if you have microsatellite stable or low or indeterminate, then NCC information are really important to be mentioned in your report. So if you don't have an NCC, we've put some example wording in the guidelines that we're recommending. You state that you do not have an estimate of neoplastic cell content, and so you caveat that your results may not be accurate because there is a possibility that the NCC can be less than your validated minimum neoplastic cell content for your assay. Or alternatively, as you all know, we sometimes receive samples with low neoplastic cell content that we still test because there is no alternative. Then if you get a stable or a low or indeterminate result, you have to caveat this clearly that this results may not be accurate. This could be a false negative result. We are using minimum or neoplastic cell content that less than that validated minimum. Of course, as you all know, if you get an MSI high result in either of these situations, then you do not need to caveat that. Now, if you are getting a result of microsatellite instability high or microsatellite stable for colorectal or endometrial testing, then you must include a statement on the likelihood of Lynch syndrome diagnosis. Again, we've put some example reporting in case of MSS. We state that we recommend you to state that this is consistent with mismatch repair proficiency, and so it reduces the likelihood of diagnosis of Lynch syndrome. On the other hand, if you get an MSI high result in colorectal or endometrial, you state that this is consistent with deficient mismatch repair, and so it increases the likelihood of Lynch syndrome diagnosis. Now, another important point which we're highlighting here is that your report may include the activation or the results of additional tests, such as BRAF V600E or MLH1 promoter hypermethylation, to support the interpretation of the risk of Lynch syndrome. You have to, however, consider any relevant national guidelines or local guidelines in your specific healthcare setting. So I've put here a snippet from the NICE guidance, which we have in the UK for Lynch syndrome screening. And you can see that BRAF V600E, MLH1 promoter hypermethylation are both dependent in the flow of the screening on the MSI results. So putting a recommendation for a follow-up testing is appropriate as per these national guidance in our case. Now for MSI high or MSS in other cancers, so other than colorectal and endometrial, you may include a statement on Lynch syndrome risk. Again, you have to consider any national or local guidelines in your specific healthcare setting. Moving on to recommendation 13, for microsatellite high or MSS for any cancer. So if you get MSI high or MSS for any cancer you're testing, we say that the likelihood of response to immune checkpoint inhibitor therapy must be reported. So you have to include a statement in the results if it's MSI high or MSS for any cancer that you're testing that reflect response to ICI therapy. We have also put some suggested wording for your reporting. So if you have an MSS result, again, consistent with proficient MMR status, and it reduces the likelihood of response to immune checkpoint inhibitor therapy. On the other hand, an MSI high result is consistent with MMR deficiency, and it increases the likelihood of response to immune checkpoint inhibitor therapy. So more suggested wording in the guidelines. Then recommendation 14 focuses on MSI low and MSI indeterminate cancer. So if you get any of these results using your validated technology, they should not be reported as MMR deficient or proficient. You basically can't make a confident call in this case. You're just saying you are uncertain of the microsatellite instability status. And because of that, the comments on the immune checkpoint inhibitor response must not be reported. However, when it comes to Lynch risk, an increased Lynch risk may be reported, but you again need to consider your relevant guidelines in your specific healthcare setting. So for us, again, in the UK, I put back this snippet from our NICE flow chart. The link is here. If you do get MSI low result, then you are still going down the pathway of a possible increased Lynch syndrome risk. Then moving to recommendation 15, if you do have MMR immunohistochemistry results and they are relevant, then they may be included in the report to aid interpretation of the MSI analysis result. So again, we give an example wording here, immunohistochemistry of mismatch repair proteins found. You then put the result of the protein result that is relevant in this case, and the tumor to be deficient or proficient in DNA mismatch repair, which is consistent with whether it's microsatellite high or stable. So if you have a loss of one of the proteins and you get an MSI high, then you can link the results together. And if you don't have a loss on IHC and you get an MSS result, again, that nice and relevant and can be included. If you have, however, discordant results, then we say careful interpretation is needed. You may consider a third mismatch repair deficiency test to resolve discordance. Again, that's not always possible. And we have mentioned some biological and technical reasons for this possible discordance between these assays. However, we haven't covered all the possible causes of the discordance, so we haven't provided a comprehensive list of these because we felt they were beyond the scope of this recommendation or this guidance. So if they are relevant, if they are concordant, you can use them. If they are discordant, you have to be careful on how you report a discordancy. But again, remember that does exist for important biological and technical reasons. So in conclusion, so that was all the recommendations, 15 recommendations between technical and interpretation and reporting. We hope that we provided an educational resource with important clarification of terminologies and with a review of the common methods used in MSI analysis, including, importantly, sequencing, which is becoming more common. We also believe that we provided up-to-date recommendations that cover your methodology, cover your interpretation, and cover your reporting, importantly with example recommended wording that we thought will be helpful for clinical labs. There's also a flow chart, which we think will be really helpful because it follows your diagnostic workflows and can be used to guide the process. If you haven't done already, like I said, this is only a taster of the guidelines. I would recommend heading to European Journal of Human Genetics and downloading this document. It is open access, so everyone should be able to download it. And if you have any questions or need further clarifications on the guidance, you can email Richard or myself for any queries. Our emails as corresponding authors are within the guidance. Now, I would like to thank many people for this piece of work. Like you've seen, people have committed over 2 years for regular meetings and for working on these guidance. I can't thank enough Richard, who did fabulous work in getting all our recommendations together. And there's Liam, Angelica, Christina, Sam, Trina, Joe, Kieran, and Patricia. So brilliant people who put a lot of effort in this. The EMQN group has been amazing in supporting this, especially Katie and Mel have put a lot of hard work in getting us organized and working to timeline in order to produce the guidance. And we need to thank all the people from ACGS and EMQN who shared their thoughts within the community consultation group. And there's a special thanks to the experts in pathology and oncology and clinical genetics who helped us as well. So Ian Frayling, Gareth Evans, Michael Braun, Sir John Byrne, and Neil Ryan. Everyone have put a lot of effort, and I really hope that you will find these guidance helpful in your practice. That's the end of my slides. Any questions? Shall I stop sharing, Mel?
No, you can leave that slide. Thank you.
Okay.
So thank you very much, George, for that extremely clear and very helpful presentation and overview of the guidelines. I do encourage all the participants today to have a look at the full document for extra information and details. So we come to the question and answer session. So at the moment, we don't have any questions. So does anybody have any questions, or was George so clear that no clarification is required?
So Well, I'll just give people a few minutes if they have got any questions to raise. I just want to reiterate, thank you very much, George, for a very, very clear presentation. I just had one question, which was where's the best place to find population specific information for MSI markers? How's the best way to get that information?
So in general, I would usually use Nomad for reliable information of population frequencies. I don't know if there are more specific ones for MSI markers other than Nomad. Richard, I know you're listening to us. Would you use any other tool other than Nomad for population frequencies?
Not specifically, no. I think a lot of MSI research or selection of markers is done based on research. You just need to get lots of different samples from different populations when you're doing that type of selection.
Okay, thank you.
So we now have a question. What could be the third test if you have discordant results?
Yeah, so I think one of the things that could be done is basically if you are using fragment length analysis and you compare to mismatch repair deficiency, so actually using sequencing, that in itself is an alternative test. If you are using sequencing and you have another lab that's using a different sequencing technology, a different set of markers, so any sequencing technology that uses different set of markers is an independent and alternative test. But you do have a spread between fragment length analysis, sequencing, and of course, IHC testing. So depending on the test that you're using and the discordance with the immunohistochemistry, you can rely on another sequencing technology for MSI, or sometimes even, especially when you're looking at MMR deficiency on immunohistochemistry, you may have results that will inform you further if you've done sequencing on the patients as well, because that does also reflect MMR status.
Thank you, George. We have one delegate who's just made the comment, thank you for the clear and to the point presentation.
That's very kind, thank you.
Karam has posted that I think some NGS methods don't have the same markers as PCR MSI, which to open the door to discordance. Do you have any recommendations for this, or is it a case of validation for the tumour site?
I would say it's, so when you're selecting the markers, so we know there will be different markers. Like I said, for sequencing, you can start from a small set of markers and expand to thousands of them, which is way bigger than any of the fragment length analysis methods. So selection should be based on literature. There are many publications that recommend or detail a lot of these methods and the markers. So they will be complementary again. Some discordance may be reasoned by biological differences or technical differences, again, when you're choosing dinucleotide versus mononucleotide and so on. So we know they are different. They will be different by the fact that sequencing can be much bigger, but the validation will be the way to determine if they are suitable or not for your own assay.
Thank you. We have a question from Catherine. With MMR somatic testing becoming more prevalent within clinical trial targets of NHS somatic panels, do you think this could have a positive or negative impact on MSI status / guidelines?
That's a very good question. I think, again, with more testing becoming available and with important differences with tissues, with tumor heterogeneity, with clonality, They can be a source of clarity, but sometimes they can confuse matter further. So we know, again, from local experience with testing, as we call it commonly, tumor lynch, was testing the MMR genes using tumor material, the analysis and the interpretation, linking the gene results in a somatic setting alongside germline results for the patients when they exist, and the MSI status, or the mismatch repair immunohistochemistry. Things can get really complicated. But when things work well, they are nice and complementary to each other. So I feel that the guidance that we're providing helps highlighting that discordance can be anticipated, and there are really valid technical and biological reasons for it.
I think I'll just add as well that microsatellite instability analysis is a functional test. You are measuring the output of mismatch repair function. Whereas if you're looking at somatic variants in the mismatch repair gene, you could have VUS, you may miss variants as well. For example, if you're using short read sequencing technologies, you may miss the fact that you have maybe large rearrangements or deletions. Therefore, MSI, I think, will always have a place alongside, as George has mentioned, the complementarity that it has with the somatic sequencing of the mismatch repair genes. And again, because it is a independent biomarker that it's measuring, it, in my opinion, will always be useful.
I totally thank you, Richard. And if you look at our variant interpretation guidelines, MSI has a place in itself, that especially because Richard mentioned the VUS in these genes, having MSI results can actually add value to the interpretation of these variants.
Okay, are there any final questions from anybody?
Okay.
I'd like to take the opportunity to inform participants of the webinar that EMQN do provide an EQA scheme for microsatellite instability. And in 2025, we had 70 laboratories participating, and the scheme catalogue is now available for anyone who would like to participate in the 2026 EQA scheme.
Okay, thank you everyone again for your time, for the excellent presentation, and thank you, Richard, as well, for joining the Q&A session. We appreciate your support for the webinar.
Thank you.
Thank you. Bye.
Bye-bye.